Okay, for the third part of this lecture, it's actually only one lecture, but it's split up into three 15-minute segments for about 50 minutes altogether, is respiratory structures. And as I pointed out, what do aquatic animals have for respiratory structures? There's some primitive animals that don't really have a respiratory system. They just use some part of their body that's got a big surface area. But what you typically see is that it's gills. Gills are what aquatic animals have. And so if you live on land, then you're going to have lungs. If you live in the water, you're probably going to have gills. These gases, we're talking about getting oxygen. Of course, the other consideration is to get rid of carbon dioxide. So these gases move by diffusion. You can't push these gases. You can't pull these gases. They're just going to move down their concentration gradient from high to low. You learned about this way back in some remedial biology class about diffusion. So if you look at gills, which we're looking at, if you look at lungs, you look at skin even, there's a couple of characteristics that these share. One is there's a big surface area that's in contact with a lot of the media that they're in. And by that I mean either water or air. Big surface area. There's a lot of water going over it. In the case of sharks, skates, and rays, that's gills. In the case of us, it's lungs. And the second thing is that there's a big blood supply to that respiratory surface. If you're a fisherman, now where's the worst place to catch a fish is in the gills, because if you gill hook a fish, it bleeds like crazy because there's so much blood being supplied to the gills. And that makes perfect sense because you've got a lot of water that's got a lot of oxygen. Where do you want that oxygen? You want that oxygen in the blood. So you transport a large volume of blood to those respiratory surfaces, and then you've got oxygen and water that's just 2 cells away from the blood. So there's a big surface area and there's a short distance for diffusion. Diffusion is a slow process. So the shorter the distance and the bigger the surface area, the higher the rate of diffusion. Or in other words, the more oxygen you're going to be able to extract and get into your blood and use for metabolism. Well, if you look at the respiratory structures in sharks, skates, and rays, as I said, they're gills. So part of the gill is supportive. There's these different parts of the gill, the rays and the gill rakers, that don't have the blood vessels, and it's not where oxygen is going to be absorbed and enter into the bloodstream. And these gill filaments are these highly convoluted, it's a very big surface area, all these little surfaces by millions of times, these little finger-like projections where water is flowing over. And if you look inside one of those little finger-like projections, the lamella, it's full of blood. So here's a salmon shark, someone's holding open its gills. You can see how red that is, how much blood there is. It's a big surface area. So there's a lot of blood, there's a lot of water that flows across these gills, and it's a short distance for all that gas, oxygen, to move out of the water, move across a couple of cells, and then it's in the blood. It's in a huge amount of blood because of all the blood that's delivered to the gills. Here's a diagram that shows these gill filaments. So you've got some structural component, the gill raker and the gill bar, and if you look at the filaments, this is where all that blood is, and this is where it's a very short distance between the water that's flowing right over the top of these gill filaments. This is the functional part of the gill. And the filaments themselves, which are filled with blood, a lot of blood. And of course, if you're talking about filter feeding sharks, then they're not just using their gills to extract oxygen, they're also using their gills to to filter out food from the water. So this is a individual gill filaments, and this is where this is the site of gas exchange, where there's a lot of gas in the water. It diffuses a very short distance over a very big surface area, and then you have a lot of oxygen in the blood. Well, it's not just set up any particular way, it's set up in a very specific way, the way that water flows and the way that blood flows. And in fact, they flow in opposite directions. There's a countercurrent circulation. Now, there's countercurrent circulation in a number of different sites, a number of different systems in animals. For instance, the blood, there's countercurrent circulation in the swim bladder of fish. There's countercurrent circulation in the limbs of some animals for thermoregulation. There's countercurrent circulation in the kidneys for reabsorption of water. So this idea of countercurrent circulation is pretty widespread in animals, but in the gills of sharks and skates and rays, it works like this. You've got water flowing in one direction across these gill filaments, the lamella, and you've got blood flowing in the other direction. So as water, if you look at water flowing into the gills, as it begins, it's got the highest oxygen concentration that it's going to have. This is water out of the ocean, and it's entering into the gills. As it's moving through the gills, the oxygen concentration in this water declines as oxygen is extracted and absorbed into the gills. Now for the blood, it's the opposite situation. This is blood that's coming from the heart that has a low oxygen content. It's being delivered to the gills in the first place for oxygen to be loaded up into the blood. So this blood is flowing along from a low concentration of oxygen, and as it comes in contact with the water, it's picking up more and more oxygen, and by the time it leaves, it's got a high oxygen content. So with water, it's a high concentration to a low concentration. With the blood, it's a low concentration to a high concentration. It's a counter current. The gradient from high to low in water is moving one direction, and the gradient of oxygen in the blood from high to low is moving in the opposite direction. So this is the situation that's set up. There's a lot of words on this slide that you can look at, and you can pause this video lecture. That's the nice thing about video lectures. You can stop this and read this or write it down, whatever you want to do. This is a situation that's going on, is not the one on the left, but the one on the right. This is where the blood and the water is flowing in the same direction. This is what would happen. The water's got a high oxygen concentration. The blood has a low oxygen concentration. So as time goes on, this oxygen flows from high to low, high to low, high to low. Well, the blood is building up a higher oxygen concentration. The water is losing oxygen concentration. So as the blood is flowing and the water is flowing, the gradient of oxygen in the water and the blood are becoming more and more similar. So the gradient that's driving movement of oxygen becomes less and less, and you have this decrease or decline in the amount of oxygen that's moving from water to the blood. Well, you want to maximize the transfer of oxygen to the blood. Now here's a countercurrent situation. You've got blood that comes in with a low oxygen concentration and leaves with a high oxygen concentration. You've got water that's got a high oxygen concentration and a low. So to begin with, there's a lot of oxygen here. There's a relatively high amount of oxygen here, but the gradient is still favors the movement of some oxygen into the blood, even when there's a lot of oxygen in the blood, because this is high. In the blood, it's relatively high, but this is still higher. Whereas here, they become more and more equal. And as you move along, the concentration of oxygen in the water gets lower and lower, but the concentration of oxygen in the blood moving this direction is also lower and lower. And the blood is flowing this direction, but if you just look at the concentration, it's a lot lower here by the B than it is here at by the F. And so this Even a small amount of oxygen in the water is higher concentration than the amount of oxygen in the blood, and you have this continued movement. And so you can extract 80 times as much oxygen out of the water with this setup, this countercurrent circulation, than you can with the circulation where the blood and the water are flowing in the same direction. That is a lot more oxygen that you can take out of the blood. So here's another way of looking at where there's actually numbers on here. If you can see this, this is where they're going in the same direction. This is what happens. This is high. This is low. Pretty soon you have this equilibrium where there's not going to be much oxygen that's taken out of the water. With a countercurrent situation, the blood comes in low, the water leaves low, but this 10 is still, this is, the 10 might be over here and 10 in the blood. You know, these aren't going to line up exactly. So by the time the water leaves the gills, it's given up a lot of its oxygen, because every situation here, this is 100, that's 90, that's 80, that's 70, that's 70, that's 60. The concentration of oxygen in the water is higher than the concentration of oxygen in the blood. So you have this constant gradient driving extraction of oxygen into the blood, out of the water and into the blood, because of a countercurrent situation. So again, there's a bunch of words up here. You can read those. 80 times more efficient that way. Well, if you look at the rate of oxygen transfer over time, countercurrent circulation, then this is what happens. If you have the rate of oxygen transfer in a situation where the water and the blood are flowing in the same direction, this is what happens. It declines. If you have a countercurrent situation, then the amount of blood, or the amount of oxygen that's picked up into the blood, is relatively constant. It doesn't decline like this over time. This is where the gradient becomes more and more similar between the water and the blood. This is where the gradient is maintained, the gradient of oxygen, concentration in water versus the blood. This gradient is maintained, and you've got a constantly high rate of oxygen extraction. So again, you can read all these. You can pause this video and look, see what all those words say. Now, the species of the day is a tiger shark. There were Lauren and Kyle, I think, did their species presentation on the tiger shark. There's a lot of aspects of the biology of the tiger shark that are really interesting. They're really non-conformists in terms of how they reproduce. They don't reproduce like other members of the same family. In terms of their movements, they've got these strange movements compared to their other members in the family in terms of migration. They are capable of being equally at home in 6 feet of water and 6,000 feet of water. They've got this wide distribution. If you looked at this, you know, we've done a lot of research on these tiger sharks. They're all the way out here in the open ocean. They eat all kinds of different prey, very indiscriminate feeders. Feed on birds, feed on snakes, feed on lizards, feed on dead things, eat a lot of garbage, feed on pufferfish and eels, and they feed on whales and marine mammals, all kinds of different prey items. Anyway, tiger shark, very interesting. They get to be about 18 feet long, and one of the top 3 species that people are interested in terms of interactions with humans, getting bitten once in a while, with the white shark and the bull shark being the other 2, the tiger shark being the 3rd one. Okay, so that is about not even 50 minutes if you add up these 3 little video segments, and that is going to make up the first of those lectures. We're going to have another one on the nervous system that's going to be available for you to watch.